Welcome everyone to one of the first talks of the Neuromatch Conference of Neuromatch 3. We're excited to start off with Carsen Stringer. I got to know Carsen as we were co-organizers of Neuromatch Academy this summer. So good to see you again, Carsen. Welcome back to the Neuromatch party. Carsen did her undergrad in physics at Pittsburgh. And then she did her PhD at UCL with Ken Harris. Since then has been at HHMI first. It's a short postdoc, and now she's a group leader. Some of you may be familiar with her software. She is one of the people responsible for Suite2P, which is sort of one of the major packages for calcium imaging out there. And now in her group, she's studying high-dimensional computations in primary and visual cortex. So welcome, Carsen.
Thanks, Sean, for the great introduction. Yeah, it's a great privilege to get to talk at Neuromatch today. And I'd just like to mention too, there's a conference the week after this for underrepresented scientists, Black in Neuro. So I'd recommend checking that out too. So thank you everyone for coming to my talk. So I'm going to tell you about large-scale neural data. In particular, in mice, we're able to record many thousands of neurons simultaneously. So I'll tell you some techniques for analyzing that kind of data and the sorts of scientific questions we can answer with that sort of large-scale data. So how do we do these large-scale recordings? We use a technique called two-photon calcium imaging. And these are in mice, which express GCAMP in all of their excitatory neurons. And here's a video of what that activity looks like. So when these neurons fire, they light up, and we can record around 50,000 neurons now simultaneously at a rate of 3 Hertz. So we're able to record a large fraction of visual cortex in mice. And this recording in particular is in a mouse in total darkness, so there's no visual stimuli coming in, but there's still a lot of activity in this mouse's visual cortex. And we're going to look further into this activity in the absence of external stimuli. So while we're doing these recordings, we can also do large-scale behavioral recordings. So here's the mouse is attached to the microscope, and we can monitor its behaviors. So while we're recording, the mouse will run, whisk, groom, and sniff, and do other natural things that it likes to do. And so the first part of my talk, I'm going to be telling you about how this activity in the absence of external stimuli, this kind of spontaneous activity, is not just noise in the brain. It's actually driven by the behaviors that the mouse is performing, like whisking, grooming, and sniffing. And then I'll tell you about how this kind of noise from behaviors, it doesn't actually impair the representation of visual stimuli. We have the mouse has highly precise representations of visual stimuli in its visual cortex. And then finally, I'll briefly tell you that these representations are also high dimensional. So there's a lot of information in mouse visual cortex about the visual world. And so how we were able to answer these scientific questions, we developed a variety of computational techniques, which I'll go over in this talk as well. And so the first thing we need to do is we need to take this raw data, which I showed you. Oh, yeah, sorry. So first, I do want to mention too, this is all open source software you can find on GitHub. I'm very grateful to the open source community in Python for all these libraries that we can use to create these great tools for the community. So we have this raw imaging data, which is pixels by time. And the first thing we need to do is, what you might be able to tell is the mouse is hooked up to the microscope. It's able to run and move around. So there's going to be some jittering in the brain. So the first thing we need to do is correct the motion in this video. And we do that using a technique called phase correlation. So I've zoomed into this video here, and on the left is the uncorrected version, and on the right is the corrected version. And so you can tell, if you look at the circled cell, you can tell this jittering would be a big problem if we were trying to determine the activity of the cell. So first we correct this motion, and then in these corrected videos, we can detect cells. And so here is that same video before. I've rotated it now and taken the max projection, so the maximum intensity of each pixel across all of time. And this is what these cells look like now in that image. And one way we could segment this data to find neurons in this recording is to use an anatomical segmentation algorithm. And something we've developed in particular is called CellPose. And if you use this, you find around 700 cells in this recording. And so that's not a bad number of cells, but we have more information than just this single frame of information. We have all of time of these pixels. We have all of these pixels in time. So what we use in Sweet2P is we actually use the correlations between pixels across time, and we create this correlation map. And within this correlation map, we actually find twice as many cells as if we were to use an anatomical segmentation algorithm. And so now we detect our cells, and in these large scale recordings, we're detecting over 40,000 cells on each one of them. And then we can take each of these cells and we need to find its activity in time. So we take the sum of the pixels in each frame, and we get these traces in time here. And these cells circled here are the ones that I'm showing in this video on the right. And so we can now start to look at this neural activity. And one way we visualize this activity is in a GUI we developed for Sweet2P. And so this allows users to click on cells and move around and explore spatial correlations in their data. You can also load in behavioral traces and do a few other things inside the GUI. And this can help you start to understand the kinds of patterns of activity in your data. And another way that that you can visualize your activity is making a raster plot, which many of you might have seen before. So we're now going to take this activity and make a plot of neurons by time. This was ultimately what we wanted to get out of our calcium imaging processing pipeline, and that this is what we get. And if I sort my neurons by their weights onto the first principal components, you have activity that looks like this. It looks rather one-dimensional if I sort by this one-dimensional projection. And this activity is correlated to one-dimensional variables such as whisker movements, locomotion, and pupil area. And again, this is visual cortex activity in mice. This is spontaneous activity. There's no visual inputs coming in. So this is activity that's just correlated to the behaviors that the mouse is doing. And in this case, in particular, one-dimensional behaviors. And this is what people have shown in several previous studies, that there's one-dimensional, that neural activity is related to kind of these one-dimensional variables. But neural activity is not low-dimensional in visual cortex in the absence of stimuli. It's actually multi-dimensional. And you can reveal this kind of multi-dimensional structure by using a nonlinear embedding algorithm. In our case, we've developed one called raster map, which tries to exploit the kind of structure, the high-dimensional structure we see in neural data. But you could use t-SNE as well, or any kind of nonlinear embedding algorithm to resort your neurons, so that now neurons that are correlated to each other are put close to each other in this embedding map. And you can start to see these multi-dimensional patterns of activity in these recordings. And so the spontaneous activity is not low dimensional. There's multiple patterns of neurons firing together. And so we don't expect to be able to explain this activity with one-dimensional measures of kind of the arousal of the mouse. And so we move to multi-dimensional predictors of the mouse. In particular, we use the whole face of the mouse in order to predict the neural activity. And so we take this face movie and we're going to basically replace each neuron here with its prediction using the face. So each neuron here has been predicted by the face in this raster plot here. And so we can see how well our prediction is, how close our prediction is to the real data. And if you kind of like squint, they look very similar. We're predicting kind of the low dimensional features of this data, but we're also predicting kind of smaller subgroups of neurons that are coactive together, maybe like this group here, and also this group of neurons here. We're able to predict these different patterns of activity in the mouse brain in the absence of stimuli. So I'll go through the computational techniques we use to do this prediction for the data in visual cortex. So we take this movie, which is pixels by time, and we can take either the raw movie or we can take the difference between frames. We get pretty similar predictions in both cases. And we take the principal components of this movie, and this gives us what we're calling mouse eigenfaces, which correspond to different movements of the mouse's face. And then we take these eigenfaces and we project them onto the movie, and this will give us these traces X in time, which correspond to the different ways that the mouse moves its face in time. And then we can take these predictions in time and predict the neural activity. And that gives us we have this prediction problem now from face PCs to neurons. And now we have many hundreds of face PCs. In this case, we usually take 500, and then we have many thousands of neurons. So we want to regularize this prediction somehow. And in this case, we use a technique called reduced rank regression. And this is a technique that kind of creates a funnel from your inputs to your outputs. So rather than having a full rank regression matrix where you have a 500 by many thousand dimensional large matrix, you funnel your input so that this input X gets multiplied by this B, so that maybe you say there's 32 components. There's this rotation of X into this 32 dimensional vector that then is used to predict our neural activity through this multiplication of A. So we're able to constrain our prediction space to a lower dimensional space. And this also gives us the ability to quantify the linear dimensionality of our prediction space. And so on the next slide, I'll go through how this prediction varies as a function of the number of components we use. And also we're going to predict not just visual cortical activity, but whole brain activity. Nick Steinmetz did these great recordings where he put 8 neuropixels probes in the brain simultaneously. And he was able to record from around 3,000 neurons across the entire mouse brain. And so we're going to take these large scale recordings, which we've also sorted by raster map so that we can see some of the structure in the recording. And we take this neural activity and we predict it from the movements of the mouse's face. We get something that looks like it's capturing kind of many hundreds of neurons that are coactive together. And again, we're also able to capture some of these higher dimensional features of how the neurons vary. So now we're going to take what I was telling you about this funnel. We can say, let's take only maybe one predictor from the face to the neuron, see how well we can do. Take 4, take 16, take 64, and see kind of the linear dimensionality of this prediction of this representation of behavior in the neural activity. And what we find is that the variance explained of the neural activity peaks around 16 components, which suggests that there's around a 16-dimensional representation of these behaviors in visual cortex and also all these other brain areas. And so this plot is showing various brain areas in different colors. And so thalamus, striatum, frontal, and sensory motor cortex are kind of more conventional motor areas. And these are the ones with the most variance explained, above 40%. And then visual cortex and hippocampus, for instance, less kind of motor related areas, they around 30% of their variance is explained. So in summary, this neural activity in the absence of stimuli is not just noise. It's related to what the mouse is doing on a moment-to-moment basis. And the next thing we wanted to ask was, okay, we're explaining around a third of the variance. Can we explain more by capturing even more of the behavior? So we're able to, as I showed on one of the first slides, we're able to capture the behavior of the mouse on many different cameras. And so we have the face camera, the front camera, a side camera, and a back camera. And we can see how well can we predict from these different camera angles of the mouse's body. And what we found was that these views that are on the face of the mouse predict more variance than these views that are on the side or the back of the mouse. And now this doesn't tell us whether or not there might be information, for instance, from the side camera that's not contained in these face cameras that's also predicting neural activity. So what we did next was we predicted from all of the cameras together, and to see if we can do better by combining, for instance, these side and back camera views with these face cameras. And when we combine these cameras, we do a little bit better, maybe a few % of variance explained better, but not substantially better. So we're still explaining around a third of the variance. So this suggests that a majority of the information in mouse visual cortex, and in this case, sensory motor areas, are explained by the facial movements of the mouse. So this suggests you basically, to capture kind of these behavioral patterns of activity in the mouse brain, you just need a camera on the face of the mouse and you'll capture most of this variability. Now, does it matter what view you put the camera at? So we looked in visual cortical recordings. We had a camera at 90 degrees and also at 45 degrees, and we looked at if the variance explained changed depending on what camera view you had. And we were able to predict very similar amounts of variance explained from both camera views, suggesting that that doesn't matter so much as long as you have enough of the face and in particular the whisker pad, you're going to be able to capture this variability in visual cortex with respect to the behavior. So in summary, we found that brain-wide neural activity is driven by spontaneous behaviors. And we were able to come to this conclusion using a variety of computational techniques. And now we're going to ask this question. So there are these patterns of activity in visual cortex in response to behaviors. Are they going to impair the mouse's ability to also see things in the world? Because the visual cortex is also used for seeing seeing things. So we're going to see if this kind of noise from spontaneous behaviors impairs visual coding, and the short answer is no. So we looked. So I'll explain this with a sort of toy example. So we have two visual stimuli. The same visual stimulus presented twice, and we're going to look at the activity of a single neuron that's modulated by running. So this neuron is going to be driven by the behavior of the mouse, in this case, the running speed. And so when the running speed goes up, this neuron's activity also goes up. And then when the stimulus comes in, this rides on top of this response to the running. And so if you were to just look at this single neuron, you're going to see noise from trial to trial. Even though you've shown the same exact visual stimulus, you'll see this noise. So you wouldn't be able to decode necessarily the stimulus using this single neuron. But we have many neurons in our recording. The mouse brain has many neurons it can use for figuring out the stimulus. So let's say if we have another neuron, which is anti-correlated with running. So it goes down and then it goes up when the stimulus comes in. If we average these two neurons' activity, we get something that has a similar response to both of these stimuli. So we're going to get reliable responses to the same visual stimulus if these neurons don't have any kind of biases in terms of their preferences for different behavioral components. So for instance, as long as the responses to this specific angle correspond to neurons that are positively and negatively correlated with running, positively and negatively correlated with whisking, all of these kinds of dependencies will cancel out, and we'll get the correct neural response on a trial-to-trial basis. And so this is the idea that the space of visual representations is orthogonal to the space of these behavior-driven directions. And so we can better explore that by taking a vector, the stimulus vector, which is the average neural responses to these various stimuli. We'll take, in this case, the responses to this stimulus. So in this case, neuron 1 and 2 are going to have high average responses, and then maybe neuron 10,000 will have a lower response. And we're going to project the stimulus vector into our whole matrix of neurons by time and see how this varies during periods where there's no visual stimuli and there are visual stimuli. And so when there's no visual stimuli and when there is visual stimuli, you can see the mouse is running throughout. So we're going to see behavioral patterns throughout this recording. So if I take this single projection of the stimulus vector, what happens is that this projection is basically zero when there's no visual stimuli coming in, which suggests this idea that there are neurons that are positively and negatively correlated with each of these behaviors, and then they cancel out to have on average zero activity when the stimulus isn't coming in. But then when the stimulus is on, you can see these responses here during this block of visual stimuli where we're showing various oriented gradings. And we can do this for different orientations, neurons that prefer different orientations, and you can see each of these has this basically flat response, this flat activity when there's no visual stimuli coming in, and then it has these projections have activity when the visual stimuli are in. Conversely, we can look at the behavioral representations and see if they're impacted by the visual stimuli. And what we see is that the projections of behavior in the neural space also have kind of ongoing activity throughout the recording. They don't seem to be affected by the fact that visual stimuli are coming in. So in the case of this kind of untrained mouse, just passively viewing these visual stimuli, it seems like the spontaneous behavior space is orthogonal to the stimulus-driven space, which will allow us to successfully decode stimuli, whether or not the mouse is running. So that this kind of noise won't impair our decoding performance. So if we look at we can also visualize this with raster maps. So I'll quickly say you can sort the neurons in that same recording where we have periods of stimulus on and stimulus off. And these more behaviorally driven neurons show up on the top of this raster map here. And then the more stimulus driven neurons show up down here. And what you can see is that when we stop the flash stimuli, this looks like the activity kind of goes to zero, which is the same idea. When we make this raster map, we're kind of averaging neurons close to each other so we can visualize these many thousands of neurons. And so you average these neurons that are close to each other that correspond to a similar stimulus, and their activity is going to be close to zero, just like we saw in that plot on the previous slide. And so we have this toy schematic where we have this subspace of behaviorally driven activity, which is orthogonal to the subspace of stimulus driven activity. And so we're not going to have an impairment of our visual coding by these behaviorally driven components. So I'm going to show I'm going to show an example of how accurate these representations are in spite of all this noise from behavior and other things. So we're going to turn this into a decoding task where we're going to try to decode from the mouse brain whether or not oriented gratings are greater than 45 degrees. And so this is an example where you'd have to say this one is not greater than 45 and this one is greater. And we're going to ask the neurons to do this discrimination task. And if we ask the neurons to do this, then what we find is we get a discrimination threshold around 0.34 degrees, which means 75% of the time we can decode from the neurons that 45.3 degrees is greater than 45 degrees. And this is a very small difference. And I mean, you might be able to tell from looking at these, you might have a hard time doing this discrimination. And indeed, this is a very small difference compared to how well humans can do. So if humans are trained on this same exact task, their discrimination thresholds are around 1 degree. So from the mouse brain, we can decode better than humans can perform on these sorts of tasks. So this suggests that there's a lot of information in mouse visual cortex on a trial by trial basis about what visual stimulus is coming into its brain. That doesn't mean that the mouse necessarily uses this information. Just like, I mean, for instance, humans, we're not using this precise of information. And indeed, if you ask a mouse to do this same task, a mouse has a discrimination threshold around 20 degrees. So there's a discrepancy between how decoding from neurons in visual cortex versus how well a mouse can do this task. So it's an open question, and we kind of explore some of these possibilities for what this discrepancy is coming from. So I recommend looking in the paper if you're interested. But the punchline is that the information isn't lost in visual cortex. It's there. It's just a question of how the mouse uses visual information to guide its decisions in the world. So to sum up that point, that part, I was talking about whether or not we have a problem in a sense that we've seen these behaviorally driven representations all over the brain. Are they going to mess with the mouse's our ability to decode stimuli in mouse visual cortex? And that doesn't seem to be the case. We're able to decode highly precisely on a single trial basis what the mouse is seeing from these oriented gratings. And so the last thing we asked, and I'll just tell you briefly, is we wanted to know, okay, these responses are accurate, but are they high dimensional? Do they have interesting information potentially about the visual world? I mean, we were just looking at orientations here. So this last study, I'll briefly touch on is where we showed many different natural images to the mice. These are images from ImageNet, from like the hawks, or the birds category and mice categories and so on, some natural categories. We showed many thousands of images, and we showed these images twice. And we wanted to know the dimensionality of the responses of neurons to this large data set. And when you want to estimate dimensionality, often what you look at is the eigenspectrum. You look at the eigenvalues of the data. But these eigenvalues can be corrupted by noise. For instance, if you have noise in your data, you're not going to be able to estimate the eigenvalues directly. And in our case, so you can't estimate them directly from a single set of repeats to stimuli. So you need at least 2. And so I'll show you what happens to your eigenvalues if you have noise. So let's say I have a matrix of signal plus some independent noise. If I look at my eigenvalue spectrum, so I have the true eigenvalue spectrum is in white, and I'm showing it in 2 different ways, on a log-log plot down here or on normal axes here. And as you add noise, you can see that these higher eigenvalues are going to be corrupted by this noise. They're going to be higher if you have noise in your data. And we don't only have independent noise, we also have correlated noise that comes from our spontaneous behaviors. And so we came up with a way to estimate these eigenvalues using 2 repeats of these stimuli in order to determine accurately the eigenvalue spectrum of the data. So again, I don't have very much time, so I'll just, I recommend looking in the paper if you're interested in more details, but we take the first repeat of data and we estimate the eigenvectors on that first repeat, and we project these eigenvectors into the first repeat and the second repeat of our data. And that creates these matrices A1 and A2, and we estimate the eigenvalues as the dot products of each of these components of these projections of these eigenvectors here. And this will produce a lower bound of our true eigenvalue spectrum, because we're estimating these eigenvectors on this noisy data, so we're never going to get the perfect signal eigenvector. So this will always be a lower bound on the true eigenvalues of our data. And when we basically were wondering, are we going to get a low dimensional eigenvalue spectrum, which would be only a few positive eigenvalues, versus a high dimensional one, which would be flat. In this case, a sparse code where every neuron is coding for a different stimulus would be a totally flat variance spectrum. And what we found was that it's actually neither of those. It decays as a power law. And we think that this suggests that the neural code is kind of balancing capacity. It wants to be as high dimensional as possible. But if you're too high dimensional, you end up like this case, like this spiky ball, where if you move a little bit in stimulus space, a completely different neuron is coding for that stimulus that might be very close in stimulus space. So having this kind of power law decay where you have some principal components, these top eigenvectors, which have more of the variance, allows you to have a more smooth representation of your stimulus space, while still also having you have these higher eigenvectors, which can encode these finer details of the space that you might want to encode. So in summary, we found that these neural responses are also high dimensional. So this suggests that the mouse has a lot of information about the visual world in its visual cortex. And it's really an open question what it does with this visual information, how this visual information interacts with the behavior, these behavioral representations of visual cortex. So that's what we're working on, is trying to understand these neural representations of behavior. Also, we're trying to determine the visual features that visual cortex is computing to guide behavior, and working on data-driven models of cortical activity. So I'd like to thank the people who've been working in my lab on these kinds of questions this past year. They're shown here, and they're applying to grad school soon. So just keep them in mind. And also, I'd like to thank my long-term collaborator, Maris Pakitsaru, and Mihaly Mihalos in his lab, who did many of the recordings I showed you today, and also my collaborators at UCL. Next time it's Matteo Carandini and Kenneth Harris. So thank you all for your time. I'm also looking for postdocs and students to work with me at Janelia. So feel free to get in touch with me. Thank you, everybody.
Well, that was great, Carsen. Thanks so much. I think I certainly really appreciated it, and I suspect that our audience did as well. So we had 200 people on the call, so that's pretty exciting. So let's go through some of the questions. Folks, please put your questions in the chat. Sorry, not in the chat, in the Q&A box, however you do it. There's a button at the bottom that says Q&A, and you can type in questions. And then you can vote for other people's questions as well. So have a look, and we got about 15 minutes of discussion that we can have. So maybe we'll start just with the most upvoted questions. So Matteo, who you know well, asked about the superiority of Sweet 2P versus CellPose. Let's see, I just lost it. Okay, here we go. I found it again. About finding the neurons that don't fire. I guess the idea being that if you're looking only at the functional, the activity correlations, that you're not going to find the silent neurons. So any comments about that feature?
Yeah, that's a great point. Yeah, and I think especially a lot of people are recording across time now with calcium imaging, and they want to have all the cells detected, whether or not they're silent in that recording. And so I think what we're thinking about is seeding Sweet 2P with the CellPose segmentation and trying to find more cells in that way. And also potentially giving the user also all the cells from CellPose. So that's a future kind of direction in Sweet 2P for sure.
Okay, cool. So let's, I'm gonna have to, I didn't have a chance to read this one, so I'm gonna just do it live here. The top question by Lawrence. Is there variation across the brain in terms of the time scale of predictive relationship between behavioral and neural data? In other words, are sensory areas more driven by moment-to-moment changes in behavior, whereas the relationship in areas in parietal frontal cortex might be more extended over longer periods of time?
I'm not sure if we have those autocorrelation plots in the paper or not. They might be in the supplement. I think the timescales are relatively similar because you have an increase in performance across kind of you have an upshift in the whole curve across from brain area to brain area. So I think there's going to be a little more. What I mean by that is there's going to be a little more modulation by the arousal component, kind of this slower component, but there's still going to be these faster components. So there might be a little bit more of the slow component, but I'm not sure if it's going to be enough to change that autocorrelation substantially. If that makes sense. Sorry, it might be in the supplement.
Okay, let's keep going. So Dan has a question. Dan, one of the big organizer folks here of NMC 3. So can you say something about what the dimensionality results might mean in terms of how the brain carries out computations or something mechanistic? He's trying to get an intuition for what it means.
Yeah, that's a good question. Yeah, so it suggests that in terms of a mechanistic perspective that there's kind of a shared input to a lot of these neurons that would be kind of these slower variance components potentially. And then these higher dimensional components might be features that are computed using smaller subgroups of inputs that neurons get. That cortical neurons might be combining kind of these inputs in nonlinear ways that would create these smaller eigenvectors. And that's just a hypothesis. We don't know. It's kind of an open question. And something I didn't mention, all the data that I talked about today is all freely available on Figshare for people to look at and try to understand too.
I have a related question. So you sort of made this comment that when you have too high dimensional of a space, then you get small changes in the stimulus direction would give you a completely different encoding. And so that makes sense for why you'd want to have these high variance components components that capture large amounts of the variance. But don't you still have the same problem for the other PCs, the PCs at the other end of the spectrum? So how is there still value of having those dimensions?
You mean like how are you still able to have a smooth representation even though you have high dimensions that have variance?
Well, but no, you have the smooth representation, I assume, because of the first set of eigenvectors. So then, is there any value in having the others? Why have them at all, right?
Right. Yeah. So it's it might be helpful for decoding in general that you have your information about the visual world in a high dimensional space, and you have kind of fine features in these smaller eigenvectors, and that might help you to do decoding problems. Like that's kind of in a deep network, you project this activity, you project this pixel space into a high dimensional space in order to turn a problem that's a very nonlinear classification problem into a linear classification problem. And so you could think that the brain could be doing something similar in that way.
Yeah, I guess, I mean, we can move on, but I guess I could it seems like if you want to preserve the invariance, which is sort of the argument for having the most of the variance in a small number of dimensions, then the other dimensions don't seem to be adding much to the problem. But anyway.
Yeah, it depends on how much invariance you want to encode, because the mouse needs to do a lot of things that aren't just related to like a classification problem, which at a certain point you can be pretty smooth and be like, the cat is in the image whether or not it's in this position or this position. But a mouse visual cortex, it needs to be doing object tracking, and these other things are probably more relevant. And so I think having precise information can be very useful for those kinds of tasks, like object tracking and object segmentation too.
So then I guess the prediction would be that in those kinds of tasks, you would be more sensitive to noise.
You might be, I think there's still a lot of neurons. So you don't need to, so as long as you know the right projection of those neurons, you can be insensitive to noise, just kind of the point of having this precise orientation tuning. In that case, we know what projection to use because we chose this one dimension that to vary in the stimulus space. But in a high dimensional natural image space, we don't really know kind of those decoding dimensions. And so we don't, you might have high accuracy in a complex space, but we don't know what those dimensions are to decode from.
Okay, thank you. Let's keep going. So Daniel Mueller, oh my goodness, Komorowska, oh my God, I'm so embarrassed. Is the dimensionality in the neural activity and behavior similar across different mice?
Yeah, that's a good question. Yeah, so we see the dimensionality of the neural activity is similar across mice. And so the power law for spontaneous activity and stimulus-driven activity, we can find that alpha, kind of how quickly it decays, and that has a pretty tight distribution across mice. And then in terms of the behavior, the dimensionality of the behavior kind of depends on how active the mouse is in the recording. And so you could look at kind of the spectrum of the eigenspectrum as well of the face PCs, and those are relatively similar across mice, but I wouldn't say they're going to be less similar than the neural PCs, I think, that the neural dimensionality that we've seen, I think.
So I'm going to actually jump to the next one. Katharina has an interesting question about whether or not your ability to do the decoding in the mice has to do with the fact that you have correlations across all of these different behavioral modes, and that might not work in humans, or you need to capture more of the full body behavior in order to decode the neural activity. What is there a consequence of the task, or sort of the lack of a task that you have here, where you're just letting the mouse sort of freely behave, run around and whisk as it wishes? And if you were in a different paradigm, would you suddenly need to have different information to do meaningful decoding in the brain?
Yeah, that's a great question. And it's still kind of an open question because I haven't looked at any task-specific data. But I think kind of what other people's work has suggested, like from the Churchland lab, for instance, is that what's in visual cortex is not necessarily going to be like, might not specifically be kind of where an arm is or what direction things are moving, but it's kind of overall arousal and what the mouse is doing. So that's the case. Actually, Nick Steinmetz's study looks at that as well. That is kind of more these overall kind of behavioral arousal and kind of, are you whisking or not whisking, and these kinds of things. Whereas maybe the specific postural information might be more in areas like somatosensory areas and motor areas. But it's still definitely an open question.
So thank you. So Hajer asked whether or not you could use these kinds of approaches to do, I guess it would be prediction of fMRI data rather than of neural data. Has anyone looked at this? Have you thought about it?
Yeah, I run raster map on fMRI data, and it looks interesting. I've looked at like kind of the power law of fMRI data. And I can't remember it off the top of my head. So you can use, I mean, it is also a multi-dimensional space, a high-dimensional space. And so you can use the same kinds of techniques in terms of nonlinear embedding algorithms and stuff like that. I'm not sure in terms of facial movements, if you have that kind of information to use that for fMRI. I don't know.
Okay, next question is, was the finding that the orofacial movements were more predictive than other body movements, I guess in terms of which cameras you're using, was that true only in the visual cortex, or did you see that when you were looking at the whole brain data?
Yeah, so that was, we did several recordings there. And so some of them were in visual cortex, some were in somatosensory areas, NV1, and then some were in motor areas. And we found across those, all of those different recordings that the face was always more predictive. But again, this is a passive mouse that's not doing a task. So if it was doing a task, maybe like we were talking about with these arm movements, maybe then you would see different correlates in these brain areas that are more motor related potentially.
I actually had a related question. So I thought it was interesting. You said that the most important thing about the camera view was that certain features of the face were present in the view. And did you consider taking those videos and degrading them systematically, either in terms of just reducing high frequency information or limiting it to very specific regions of the image, and see could you tease out what were the aspects in the video that were most important for predicting the neural data?
I don't think I've done it as systematically as you're describing, but I've at least looked at the whisker pad versus the rest of the face. And the whisker pad contains around 75% of it has about a performance of 75% of the best possible performance. So it has most of the information. That's
really interesting. I mean, it gets back to our previous question that, there are, at least in these spontaneous movements in rodents, there seems to be a high level of correlation. I'll just mention really quickly that Elizabeth Hillman here at Columbia, I've been talking to her about some of her data, and she sees the same thing, that you can predict like a lot of the behavior of the full mouse just looking at whisking movements. So that's really interesting. Okay, so Byron has a question. Did you notice that the dimensionality and the reduced rank regression technique varied depending on the sampling frequency of the data? So 3 hertz, 2P data was at 3 hertz, but the Steinmetz spiking data, you can sort of arbitrarily choose your rate.
Yeah, we didn't actually see a difference in that. And that actually, so we did do smaller bins, like 100 millisecond bins, and I think it saturated around the same point. You can look in there's a figure on that in the supplement, but I'm not sure if it's exactly the same analysis that you're describing. Like, I'm not sure if it's exact same curves, but yeah.
Okay, so we have only 2 minutes left. Maybe we'll do one more question and then we can sort of wrap it up. So one of our participants, a Zoom test, asks, do you think neurons in visual cortex having far more information for decoding than the mouse actually uses might be a general finding in other regions? Yeah, this is really interesting.
Yeah, it would be really cool to see if other people find similar results in other areas. I would expect so in primary auditory and other regions. I would guess. Mouse performance is never 100% on any of the tasks we train them on. These like kind of laboratory tasks, they're never getting to 100% performance. So it's an open question of like, do they not have the right learning rules kind of to learn these tasks? Are they kind of better at one-shot learning and other things that are more behaviorally relevant? Yeah, it's an open question.
So maybe I'll just follow up on that. I mean, you said in the paper, you kind of speculate about what might be going on and why the mouse's performance is so much poorer than the neural decoding capability. Can you just give us just a couple of ideas and as take-home points here?
Yeah, so one hypothesis we had was that it could be that it's learning, the way that it learns is not optimal. So like maybe it finds a couple neurons in the population to pay attention to, and then that would degrade its performance to mouse level. Or it only uses a couple of trials to create its discrimination boundary, how it decides to do the task. And that similarly will degrade your performance too.
So he's going to sort of lump those together, saying there may be some inefficiency in how the learning is conducted.
Yeah, that's one hypothesis.
Okay, well, I think that's it for us. So thank you so much, Carsen.
Thanks everyone for coming.
Everyone, yeah, thanks everyone for coming. And hopefully we'll see you all at more talks this week.